So this chapter is on cellular metabolism and how different reactions and the types of reactions that happen within the body. All right, so when we say metabolism, it has to do with, again, all the reactions that are occurring within the body or within the cell. And we have two types of metabolic reactions, and those are anabolism and catabolism. The main difference between the two is that with one, you are building molecules, and the other, you are breaking molecules down. So with anabolism, you are taking molecules and putting them together to make a larger molecule. So you're building molecules. So we look at the example that's at the bottom. If the reaction was going in this direction, meaning we're taking two monosaccharides and making a disaccharide, that would be an anabolic reaction. Whereas with catabolism, we are taking bigger molecules and breaking them down. So if we were talking about the reaction going in this direction, we're taking a disaccharide and breaking it down into two monosaccharides. This would be a catabolic reaction. Okay, so that's the main difference. We're going to go over examples of anabolic reaction and catabolic reactions. So for anabolic reactions or anabolism, again, this is where we are taking two smaller molecules and we're making a bigger one. The example is dehydration synthesis. Now, if we were to take dehydration synthesis, kind of break that word down. Synthesis means that we are making something or to produce something. So we already said that anabolism is where we are forming larger molecules. So dehydration synthesis means that we are producing or making larger molecules. Dehydration, when you think about that, you may think about water. Okay? Or having a lack of water, or maybe water being removed or something. You're thinking about water. And so whenever we're going through an anabolic reaction, the two smaller molecules are going to not only come together to produce the bigger molecule, in this example, monosaccharides producing disaccharides, but they're also going to take some of the molecules that are within them and it's going to produce water. So anytime you see molecules that are coming together, they're producing something, and water is produced, that means we have an example of an anabolic reaction. On the opposite side, we have a catabolic reaction. An example for this is hydrolysis. Again, hydro, we were to break this word down, means water. So we have water that's going to be involved. But lysis means to break. And so if you break that word down, hydrolysis, that tells you that we're breaking something down. And we've already said that with catabolic reactions, we are breaking down a larger molecule, in this example, the disaccharide, and making smaller molecules. But we also know that water is being used because of the example or the name hydrolysis. That means that we are going to be using water. In this case, the disaccharide uses water to help break it down into two smaller molecules. So it's going to be using water to split whatever those complex substances are. So this will be the opposite of dehydration, synthesis. Synthesis, we're making. Hydrolysis means we're breaking. So we want all these reactions to happen, and we have all these types of reactions, but they can't just happen without something kind of controlling the rate of them or helping them to happen. Some reactions need a bit of help to allow them to go faster. They may need a little bit of help to break them down. And those substances that we have in our body that help to control the rate of these reactions are called enzymes. And they're types of proteins that we've covered so far. But we're going to talk about enzymes and how they kind of control the rate of these reactions. So when we think about an enzyme, one of the main functions of an enzyme is to speed up the reaction so it can happen a bit faster. That's why we call them catalysts. They help to speed up the reaction. But even though they're speeding up the reaction, they're speeding it up. So even though they're speeding up the reaction, they're lowering the activation energy, which means the amount of energy that is required for the reaction to start. So it's needing less energy to start the reaction because of the presence of the enzyme. And every enzyme is going to be specific to its particular substrate. And one enzyme will connect to that one substrate, and it won't be able to just connect to any other substrate because they are very specific. That's one of the characteristics of an enzyme. It's very specific. For instance, the enzyme lactase breaks down the sugar lactose. So this enzyme is very specific to the sugar that it breaks down. Another thing that we need to know is that the enzyme has, or a couple parts of the enzyme, one part of the enzyme that we need to know is the active site. The active site is going to be the area that the substrate binds to the enzyme. Now the active site is going to be part of the enzyme, but this is going to be what the substrate binds to. In this case, we have two molecules here. They're going to bind to the active site of the enzyme, and it produces this product. So again, substrates are going to be very specific to the enzyme. Also, another thing that I did not mention on this slide, but you probably know this, but anytime you see ASE, that's kind of a clue that we are talking about an enzyme. Like sucrase, protease, kinase, those are going to be enzymes. So ASE usually is at the end of enzymes. And so our enzymes, we want them to help, again, speed up reaction. We want them to help to lower the activation energy and be very specific for their substrates, but there are things that could possibly affect the way that they work. For instance, if it's too warm, if the temperatures are too warm, then the substrate may not be able to break it down like it needs to. If it's the pH is too high or too low, then the enzyme may not be able to work like it needs to. And those things that will affect the enzyme could possibly inactivate the enzyme. And so if you raise the temperature, if it's supposed to be at a certain temperature, you raise the temperature, the enzyme may become denatured, which means that it's just not going to work. Or maybe even alter the structure of the enzyme. And if the structure of the enzyme is altered, then the active site of the enzyme is altered, which means that the substrate cannot now bind. We also have some other ways to control reactions. And the pathways or reactions within the body are not usually just this one-step process. Some are, but many are not. And whenever you have an enzyme, for instance, enzyme A, that binds to substrate 1, it's going to create a new product. So this is the product, substrate 2, that comes from the reaction of these two. But this substrate or this product could act as the substrate for the next enzyme. And so this enzyme will bind to this, or that substrate will bind to that enzyme. It will produce a product, and that product could then be the substrate for the next enzyme. And that could continue to happen until we get what we really want, which is our final product. And this could also be used as a negative feedback mechanism, where if we have too much of that product, it could slow down the substrate 1 from being produced or enzyme A from working. If we have too little of that product, it could cause them to work better and connect better and bind better. And this part here at the end is what I was referring to. Usually the substrate and the enzyme are going to be very close in name to each other, like lactase and lactose. And then the enzyme is going to usually end in ASE. For instance, protease breaks down proteins, lipase breaks down lipids. And this is what I was just referring to in the last slide, whereas the final product could kind of influence what's happening on the other end of this whole metabolic reaction. And that first enzyme could be what we call a rate-limiting enzyme, which means as that enzyme works on the substrate, it's going to produce the other substrate, which will then work on its enzyme and produce this other substrate. And then that product, if we have too much of it, it could start to inhibit this from being produced, or slow it down from being produced. Or it could do the opposite, where if we have too little, could promote that rate limiting enzyme. And that is negative feedback. If y'all remember, negative feedback is usually going to be the opposite of the stimulus. So if we have a lot of something, it's going to go back in the opposite direction. So if we have a lot of that product, it's going to slow down that rate limiting enzyme because we don't need to continue to make more of that product. Or if we have too little, it's going to speed it back up so that we can get enough of that product produced. So in order for all of these reactions to happen, most of these reactions to happen, we have to have a bit of energy that's going to be produced. And the energy for our cells are produced mostly through cellular respiration. We have other ways and other mechanisms that help to produce a little extra ATP, but the primary producer of our energy within our bodies and within our cells will be through cellular respiration. And that is going to form chemical energy. And we have all these other types of energy, but the one in which we need for the reactions we're referring to will be using chemical energy. So the chemical energy that we use is ATP, and we may already know that it stands for adenosine triphosphate. Adenosine because it is made up of adenine and ribose. That's the adenosine part. Triphosphate means that it has 3 phosphate groups. Okay. And a very important point to note is that the second and third phosphate groups, this is where the energy is stored. And so whenever energy is released, it is because the bond between the second and third phosphate group has been broken. And that's where that energy is stored. So we also have a molecule called ADP. When ATP, remember we have adenosine triphosphate, when it loses that last phosphate group, it turns into adenosine diphosphate. That means that it now has 2 ATP. And ADP can then be converted back to ATP. Remember, ATP is 3 phosphate groups, whereas ADP has 2 phosphate groups. So the way that it can be converted back to ATP is by simply adding another phosphate group to it, and now we have ATP. And the cycle continues over and over depending on what's going on or what's needed. So this next section, we're going to go into cellular respiration, which is the mechanism that we create most of our chemical energy. And cellular respiration is going to be happening to a molecule of glucose. And we have 3 steps or 3 reactions that occur. And they are glycolysis, the citric acid cycle, and the electron transport chain. Now, glycolysis, if you notice beside it, it says that it's anaerobic. Anaerobic, which means that it does not need oxygen. So it occurs without oxygen. And aerobic means that it has to have oxygen. So these last 2 steps occur with or in the presence of oxygen. And that's the actual order that cellular respiration occurs in. And we'll talk about each one of those. So this looks very similar to the diagram that we were just referring to on this slide. Now we're just seeing it as a cycle, because usually that's how it happens. We have this reaction that's going to start. We have a substrate. It's going to react on its enzyme that will produce a product that will be the substrate for this enzyme. And so it's going to be just one continuous cycle. And a part of cellular respiration that is a cycle will be the citric acid cycle, which is that second part of cellular respiration. So this diagram shows us the 3 parts of cellular respiration. We have glycolysis, that's here. The citric acid cycle, that cycle that we were just talking about. And then the electron transport chain. And this whole thing, this picture is showing a cell. But this inside part here, this more orange part, that's going to be the mitochondria. And this part is just the cytoplasm of the cell, or the cytosol of the cell. You notice glycolysis occurs in the cytosol. That's important to make a note of. And the citric acid cycle and the electron transport chain are taking place in the mitochondria. So we're going to start just talking about just basic products of cellular respiration, things to kind of look out for as we go through these steps. Basic products of cellular respiration: carbon dioxide, that will be a byproduct, water, ATP, and we're always releasing heat energy. I've already mentioned the terms aerobic and anaerobic. Anaerobic respiration is going to occur in glycolysis, whereas the other ones are going to be aerobic respiration. So the very first one, glycolysis. Glycolysis is the breaking down of glucose. So remember, I just said that lysis is to break down. So glycolysis is the breaking down of glucose. And you may remember that glucose formula is C6H12O6, which means that we have 6 carbon molecules. So if glucose is being broken down, then that's going to result in two molecules that have 3 carbons. Okay? So if we have glucose that is a 6 carbon molecule, when it is broken into 2, we're going to have 2 molecules that have 3 carbons. And those molecules that are formed are pyruvic acid. And we just saw a picture a few slides ago here that glycolysis occurs in the cytosol. And we've also talked about it being anaerobic, which means that it does not have oxygen that's required. Another thing to note is that in glycolysis, we have 2 ATP that are yield. But I want to make a note that this is 2 net ATP. 2 net ATP. Now, if any of you all had a job at any point in your lives, you know what the word net means, right? So, for instance, if you have gross of $2,000, your net pay may be $1,500 or $1,000. So the net is what you take home even after you've put something in or something is taken out. Okay, so this is what you leave with. So after glycolysis, we leave with 2 net ATP, but there are other things that go on, which we'll talk about here. So the very first thing that happens in glycolysis is that glucose molecule, that 6 carbon molecule, is going to be phosphorylated, which means that we're just going to add phosphate groups on the ends of glucose. In order to do that, we have to use 2 ATP molecules. So this is kind of like us investing. So we're kind of investing. These are invested molecules of ATP. We're putting this in. For instance, if you're starting a company, you may have to put in $2,000. Okay, so we've invested 2 ATP. The next step is the splitting of glycolysis, which is what it gets its name for. We have that 6 carbon molecule of glucose is going to be split down the middle. And now we have our 2 molecules here that are 3 carbon molecules. Those molecules will then go through other series of reactions, but in the end, we're going to have our two pyruvic acid molecules that have 3 carbons. So we have 3 carbon molecules that came from the 6 carbon molecule of glucose. So in the end, we have our 2 pyruvic acids, and we also have 4 ATP that's formed. Now, if you remember just a few slides ago, we said that there are 2 ATP molecules that are produced. But I also said that there are 2 net ATP molecules that are produced, which means, if you remember from step 1, we invested 2. In step 3, we received 4. So that means that we have a 2 net ATP that have been received or gotten out of glycolysis. The example again, if we invested $2,000 into a business and we receive $4,000 back, our profit or our net is now 2. Okay? So make sure you know the difference. If we have 2 invested and we get 4 out, then our net ATP for glycolysis is 2. Some other things that are formed are NADH and some hydrogen ions. And those 2 things will be taken all the way to the end, specifically the electron transport chain, where we're going to need a lot of high energy molecules to make most of our ATP. So, for glycolysis, we're splitting glucose into two pyruvic molecules, pyruvic acid molecules, and both of those have 3 carbons. And in the end, we create 2 net ATP. So again, anaerobic respiration, which means we do not have oxygen, glycolysis is going to occur. If there is oxygen, which means in the presence of oxygen, those NADH molecules and those hydrogen molecules or hydrogen ions are then going to be transported to the electron transport chain, and there will be a series of reactions that will happen at that spot, which we'll talk about when we get to that point. However, if there's an absence of oxygen, that means we won't have our final electron acceptor, because oxygen is going to be the final electron acceptor. Maybe a good note to have. So since we don't have oxygen as our final electron acceptor, those molecules of NADH and hydrogen ions are going to then help to form lactic acid. And lactic acid will then decrease the production of ATP, and we basically have the 2 net ATP that are produced. And the reactions will then stop because they won't go into the citric acid cycle and the electron transport chain, because remember, for those 2, we have to have oxygen present. So let's go on into our aerobic respirations, aerobic reactions. And so when we do have the presence of oxygen, that pyruvic acid molecule that was produced at this step is going to move on into the mitochondria. I'm going to go back to this slide here. So this is our pyruvic acid molecule that we produced from our 2 pyruvic acid molecules that we've produced from glycolysis. They are now going to enter into the mitochondria and start the citric acid cycle and the electron transport chain. So the very first thing that's going to happen, there are a series of reactions, and I'm not going to ask you everything that happens within and in all the different precursors, but what you do need to know is that those two pyruvic acid molecules are going to be used to produce a coenzyme, and that coenzyme is called acetyl-CoA. It's going to produce a coenzyme, and that coenzyme is called acetyl-CoA. And that acetyl-CoA is going to go through a series of reactions, and I'll show you all the picture in a few. But in the end, we're going to produce, at the end of all of our aerobic respiration, which includes the citric acid cycle and the electron transport chain, we're going to produce 36 ATP. In addition to carbon dioxide and water, which were the other products of respiration. So looking back at this picture, this is our pyruvic acid molecules that are being produced. But remember, we have two of these. Important point to note, because we have that one glucose molecule is broken down into two pyruvic acid molecules. Okay? Now this picture is showing what happens when we only have one. Okay, so I'm going to erase two so that you won't get confused, because what's on this slide is in reference to what happens to a single pyruvic acid molecule. So a single pyruvic acid molecule will enter the mitochondria. It will go through a series of steps. Remembering one of those steps will be to produce acetyl-CoA, so remember that part. I will not ask you all of these precursors that it goes through when it goes through the citric acid cycle. You won't have to know every name that's here, but you do need to know that those pyruvic acid molecules form acetyl-CoA, which is just a coenzyme. And some of the byproducts, we'll talk about that in a few, okay? But what I want you to know and want you to remember that one pyruvic acid molecule that has been produced, one of those molecules will go into this cycle. And once it goes into this cycle, this is what's produced. So each pyruvic acid molecule that was produced from glycolysis is going to produce 1 ATP, 8 hydrogen atoms, and 2 carbon dioxide molecules. So, the question that I like to ask here, you may want to pause so that you can kind of think. Pause the video for a second. We just said that one pyruvic acid molecule is going to enter into the citric acid cycle and is going to produce 1 ATP. The question is, how many ATP are produced in the citric acid cycle from one glucose molecule? And I'll repeat that question. How many ATP are produced during the citric acid cycle from one glucose molecule? And if you need to pause at this point, I would probably recommend so that you can kind of think about that question. How many ATP are produced for each citric acid cycle from one glucose molecule? So the answer to that would be, hopefully, 2. Now you're probably saying, well, 1 is right here. But this is saying for each turn of that citric acid cycle. And for each turn, we only have one pyruvic acid molecule that comes through. So that one pyruvic acid molecule will come through, it'll go through this cycle, and we'll have one ATP that's produced. But remember, glycolysis will produce two pyruvic acid molecules. And so since we have two pyruvic acid molecules, one will go through, produce one ATP, and then the next will go through the citric acid cycle and produce another ATP. So from one glucose molecule, we have produced two ATP from the citric acid cycle. So we had to just kind of make a note of what we've done so far. I'm just going to write this like off to the side here. So in glycolysis, and this is all net speaking, we have two net ATP that are produced. And this is all from one glucose molecule. So glucose, a single glucose is going to produce all of this. So it's going to produce two net ATP in glycolysis, but with the citric acid cycle, we're also producing two ATP. And the reason why we're doing two ATP is because, remember, glycolysis has two pyruvic acid molecules that it will form. For each pyruvic acid molecule, they're going to enter the citric acid cycle once. Once they enter the citric acid cycle, once the first one enters, it's going to produce one ATP, and then we have a second pyruvic acid molecule that's going to go through, and that will produce the second ATP. Okay? Now you may be wondering, possibly, we just said that in the end, we're going to have a total of 36. But at this point, we've only produced 4 ATP from a single glucose molecule. So that means that this next step, which is the electron transport chain, has to be responsible for producing tons of ATP. So, the very first step. So if y'all remember, we were talking about how hydrogen ions are produced, NADH, all these high energy molecules and electrons are being produced in the different parts of respiration, glycolysis, and the citric acid cycle. And all of those high energy electrons are going to be transported to the electron transport chain, which is basically on the inner membrane of the mitochondria. Those high energy molecules are then going to be transferred to an enzyme called ATP synthase. And let's pause there. ATP synthase, what did that look like it may be doing? So ACE, we know it's an enzyme, that's the ACE part, right? Synthase may sound like it's synthesizing something or making something. What do you think it's making? ATP. And so ATP synthase will be using those high energy electrons to phosphorylize or add a phosphate group to ADP to produce ATP. And we'll continue to do that and continue to do that a lot because we have a lot of those high energy electrons that have been transported to this point. And we're going to make a lot of ATP. We also have oxygen that went into respiration. Remember, we had to have oxygen to even get to this point. Oxygen is going to be our final electron carrier. So it's going to take that final electron after all the ATP has been made. That final electron will then help oxygen form water. And remember, water is one of the products of respiration. So just an overview of cellular respiration. The first step, glycolysis. We have two net ATP that's produced. And it's also going to produce 2 pyruvic acid. But each will enter separately. So from one turn of it, we're going to produce acetyl-CoA, and then it'll produce 1 ATP. The second will come, it will produce another ATP. So it produces a total of 2 ATP. And then all of those high energy electrons that are produced in glycolysis, produced in the citric acid cycle, will make its way to electron transport chain, which will then give its energy to ATP synthase so that it can make all of these ATP, specifically 32 to 34 ATP, and also water. So we have 2 ATP plus 2, which is 4, plus 32 or 34. And that's why you usually see that cellular respiration will form between 36 to 38 ATP, because this could possibly vary. So at this point, I'm going to have 2 questions here, or 2 things to kind of think about from what we've covered so far. I would recommend pausing the video here and trying to answer these questions on your own, or these prompts on your own. And if need be, go back through your notes. If not, awesome. I'm going to pause for just a few seconds, and then you can also pause the video. Okay, so what I want you all to do is to write your answers to these 2 questions in the comment section of the video, and we can talk about these in class. We also have some matching that I would like you all to attempt to do. Again, pause the video for a second or for a few minutes. I'll also pause here if you need to. Attempt to do as much as you can without the use of your notes, but if you need to look back, do so. And this will also be something we'll talk about more in class. All right, so this next section is on nucleic acids, which we know are DNA and RNA, and protein synthesis, and how they're kind of related to each other. So, nucleic acids, the 2 nucleic acids we have are DNA and RNA. We know that DNA is going to be deoxyribonucleic acid, and it's going to be our genetic material to help us look the way that we look, because it holds the message that we need to synthesize all of the proteins that we have to help us look the way that we look. And it helps to store information on how to create proteins, not only structural proteins on how we look, but enzymes and antibodies and just a host of things within the body. So just first, we're going to talk about a few terms and just some terminology. When we say genetic information, that's what DNA stores. This is kind of the step-by-step instructions on what we what the cells need to process in order to create us, okay? And we're talking about a gene. If we were to take DNA and kind of stretch it out, and we were to section off a portion of DNA that has the information for one particular protein, that's going to be a gene. For instance, if we're talking about the protein hemoglobin, so there's a part of the DNA that specifically codes for hemoglobin. That will be the gene for hemoglobin. If we were to just cut that part of the DNA out. And then genome is everything. So from start to finish, all the chromosomes, just everything. Exome is the part of the genome, remember genome is everything, that specifically codes for proteins. So yes, we do have parts of the genome that is not responsible for coding proteins. And then gene expression is what it kind of sounds like, to express those genes, to make them come alive so that we can see them, so that they're produced. And it also controls how much of a protein is produced and when they're going to be produced. So, going back a little bit into the structure of nucleic acids, we're going to talk about, remember we said that the building block of a nucleic acid is the nucleotide, and the nucleotide is made up of a sugar. And if we're talking about DNA, the sugar is deoxyribose, also a phosphate group, and a base. And those bases are going to be specific to DNA, very similar to RNA, but there is one change. You all may have been taught before that DNA resembles a twisted ladder or a double helix, because it is a double-stranded molecule and is twisted on each other. And so if we're drawing a DNA molecule, I'm not the best drawer, but if we were to draw a DNA molecule, the backbone, and we have these things that kind of connect. I guess that did come out pretty good. The backbone of the DNA molecule is going to be the sugar phosphate. So that's this portion. And the part that's in the middle, those are going to be our bases. And it says it's 2 strands. And this will be a strand here. Like this is one strand, and then the other strand is here. And they hold those together. They're held together by specific types of bonds, specifically hydrogen bonds. And I have a question here: which bases bond together? Hopefully you remember. We have adenine, thymine, cytosine, and guanine. And A will bind with T, and C will bind with G. Or adenine binds with thymine, whereas cytosine binds with guanine. And this is just showing an illustration of the double-stranded DNA molecule, where the backbone is the sugar phosphate, and then the bases are going to be bonded or be bound together by hydrogen molecule or hydrogen bonds. Also, as far as the structure of DNA, I've already mentioned that it's a double helix and what it's made up of. We also said that the bases are adenine, guanine, cytosine, and thymine. And when it twists, actually twists around these proteins called histones to help kind of hold it together to make chromosomes. So We know that there are times when we have to have more DNA, for instance, when we're going through the cell cycle or interphase, this S phase of interphase, and we're trying to make more DNA. And so we're going to talk briefly about DNA replication and what happens. And so we know that we have this double-stranded molecule, and we have bases that are bound together. And I do have a little space here, but they're actually connected. We have those bases that are bound together. The first thing that's going to happen is those hydrogen bonds that's between the two bases, they will break. So it's going to break that apart. And so because they're breaking apart, now we don't have the strand that's bound together, and so those strands are going to start to unwind, they're going to start to separate. So now we have two separate strands. And where those two separated strands are, I don't know if I'm this good of a drawer, but let's see. Probably not. So I'm not going to try to draw that. But where those two strands are separated, I think I may be able to. Let's see if I can try to draw it. I'm going to draw this one where they actually aren't touching, like we're close to being touched. These actually are touching. So here, where these have started to separate, we will have nucleotides that are kind of free or floating around, and they will bind to this. We'll have one that can bind to this one or this side. And so as it unwinds, it will start to have nucleotides that are free that will start to bind to it. And then you have another one that will bind to this one. And you'll have another one that will bind to this one. You have one that's going to bind to this one, and one that's going to bind to this one, and then one that will bind to this one. And so eventually, you're going to have one strand, two strands. So you have two strands where both strands will have an old strand and a new strand of DNA. So I want y'all to practice and you can press pause again. If we had a strand of DNA, and this was a strand of DNA, what would the nucleotides look like that are bound to it? What would they look like that are bound to it? Again, you can put this in the comments. We'll also go over this in class. Or we could do other examples in class. And so, hopefully you've paused it. And so the strand of DNA that will look like this, or that will bind to it, will be that strand. And this is the image that I was attempting to draw, where you have the old strand here, it's unwinding, and you have those free nucleotides that are binding to it, and we end up with two new DNA molecules. So, in addition to that, we have something called a triplet code, which is basically triplet means three. We have three nucleotides that are together, and those three nucleotides are going to represent an amino acid. If you remember, amino acid is the building block of a protein, and that's going to help to create proteins. Another term, not sure if I've said this before, but a polypeptide is just another word for a protein. Remember, peptide bonds are those that hold together amino acids. And so if you see polypeptide, that's just another name for a protein. And so protein synthesis is going to be definitely highly reliant on DNA because it's going to have the instructions or like this slide says, the master copy. But DNA always stays in the nucleus, but protein synthesis is actually going to happen in the cytoplasm. So you have to have something that's going to get the information from the DNA and take it to the cytoplasm. And that's where RNA comes into play. So RNA is going to be what's responsible for taking that information from DNA and taking it to the cytoplasm. Because DNA, of course, has, again, the master copy. So let's talk about RNA structure for a few. One of the differences between RNA and DNA is that the sugar for RNA is now ribose, whereas the sugar for DNA is deoxyribose. The other difference is it does not have thymine, but instead it has uracil. And so the base pairing in this case still is C to G, but now A is bonding with U because we don't have thymine there. So when we are trying to get the message from DNA, and RNA is the one that's responsible for getting that message and taking it back to the cytoplasm. The process of getting the message from DNA and turning it into a form that RNA can use is called transcription. Now, the word transcription comes from transcribe, which means to write. So we're just writing down the message that DNA has so that we can now do what needs to be done to make the proteins. And so the specific RNA that's going to be responsible for getting that code from DNA and taking it to structures within the cytoplasm is called messenger RNA. And so the messenger RNA is going to get that code and take it to the cytoplasm, specifically the ribosome in the cytoplasm. And the process of RNA doing that is called transcription. And so we have another enzyme that's going to help us do that, and that's RNA polymerase. And that's going to be just the enzyme that helps to speed up the reaction of mRNA copying that strand of DNA. So, steps of transcription. Remember, we have our DNA. DNA has the code. It has what we need. It has that copy, or it has the master copy. And so RNA polymerase is going to recognize that DNA strand that needs to be copied. Then DNA is going to start to unwind so that it can start to be copied. Then mRNA is going to come there, and this is all happening inside the nucleus because this is where DNA is located. And it's going to start pairing A to U and C to G. So on a DNA strand where there's a C, it's going to pair G's. On the DNA strand where there's an A, it will pair U's. Or wherever there's a T on the DNA strand, it will pair an A. And it will continue to do that until it gets to a signal where it says this is the end of the gene. Then after that, the DNA will then rewind, and the mRNA is going to leave the nucleus and go to the cytoplasm, specifically the ribosome of the cytoplasm. So, just a little bit of practice, and again, you can pause this for a second so that you can do it on your own. If this is the DNA strand that's in the nucleus, what would the RNA strand be? or the mRNA strand. The mRNA strand would be this. Remember, wherever there are A's, it will pair with U. Wherever the DNA strand is a T, it will pair with A. So this is the DNA strand, and this is the RNA. Okay? And this is just another example showing the DNA strand on the left and the possible RNA strand on the right. All right, so the next step is translation. Now, the word translate to you may mean that we're going from one language to the next. And that's exactly what we're doing here. So we've already gone from DNA to RNA, which is they're both nucleic acids. But now we're about to go from RNA to protein, which is a different language. And this is happening within the cytoplasm, specifically the ribosome that's in the cytoplasm. Okay, so the code that we have now is on an mRNA strand. They're going to be separated into 3 bases, and those 3 bases together are called codons. And so let's say, I can't remember exactly what it was on the last slide, but let's say it looked like this. We will have, this will be a codon. This will be a codon. And this will be a codon. So those are 3 codons. And then those codons will be used to synthesize a specific amino acid, and those amino acids will be joined together to create a protein. So the practice I want y'all to do at this point is, if this was a strand of DNA, what would be the strand of RNA? But specifically, I want you to also separate them into codons. Again, you can pause for a second or a minute or however long you need to. This is what you should have. And this picture here shows the different steps. So we start here with transcription inside the nucleus, and we have the DNA strand that's here, it's unwinding, and we have our mRNA strand that's being formed here. This mRNA strand, after it has gotten the message, this is transcription, is then going to leave the nucleus through a nuclear pore and into the cytoplasm. Once it gets to the cytoplasm, we'll then find a ribosome. So this is our mRNA strand, and it's on a ribosome. And remember, we said that this mRNA strand is separated into codons. So the triplets are codons. And those codons are going to code for a specific amino acid. And the amino acids are these purple structures here that are attached to this weird T structure here. But those structures are carrying the amino acid to the ribosome. We're going to talk about what those structures are. So, that T structure that I was referring to is called the transfer RNA. The transfer RNA is going to have the amino acid attached to it. And remember, I just said that every amino acid codes or is coded by a codon. So that codon tells us the specific amino acid that should be in that spot. Now the question is, how would the tRNA know to attach to the mRNA strand at that point? Like, how does it know that this amino acid goes with that codon? The way that it knows is that the tRNA has a structure on it called an anticodon, which is complementary to the codon. So for instance, if this was my mRNA strand, and my mRNA strand was AUG, GCA, this is one codon, this is another. I will have a T or transfer codon that will come in, and it would have an anticodon attached to it. The anticodon would be UAC. And so this is going to be attached to the transfer RNA. And I'm going to show you all where it's located on this picture. So if you look at these T structures, I said this is the amino acid, and this T is going to be the transfer RNA. But these structures that are at the bottom, those are the anticodons. And those anticodons are complementary to the codons of the mRNA. And so that's how the transfer codon recognizes the correct codon. And once it recognizes that correct codon, it will then continue to bring the amino acids there until it has grown into a protein. And so for each codon, again, it codes for an amino acid. There are 20 types of amino acids. We have several types of codons. There are more codons than there are amino acids, which means that there are certain codons that can, or there are multiple codons that can code for the same amino acid. Could be just one codon, or it could be up to 4 codons that code for a single amino acid. Now it's important to know that we have what we call an initiation codon that is going to be responsible for starting protein synthesis. So it will read the mRNA strand until it gets to AUG. If it does not see AUG, then that's not going to be where the protein starts. All proteins will start with methionine, which is the amino acid that is coded when we see AUG codon. We also have something that tells the strand when to stop producing or to stop making more amino acids bound or bind together. And we have 3 of those, and those are called stop codons. I know UAG is one, and there are 2 others. So let's look at a codon chart here so y'all can kind of see what I'm talking about when I say that multiple codons could code for the same amino acid. For instance, CCU, CCC, and CCA and CCG all code for this same amino acid. But UAU and UAC are going to only code for tyrosine. So it could be just 1, it could be 3, it could be 4 codons that are going to code for a particular amino acid. And again, this is our start codon, which is AUG. We have our stop codons, which is UAA, UAG, and UGA. And so that's when it knows to stop. And the way that we have several types of codon charts. The one in your book may look like this, but there are others that may look a bit different. But the way to read this one, let's say the codon was AAA. If you notice on the side it says first letter A, so you find the first letter A along the side, which is here. And the second letter is here, so the second letter is A. So we find where those two cross. So this is the second letter A, this is the first letter A. So where those two cross is in this box. And so we just find what we're looking for. Is it AAA, which is here. And AAA codes for lysine. So that's basically how you use this codon chart, but there are several types that are out there. Some are easier to use than others. So, and this is what I was just referring to about the mRNA molecule are going to have the codons and the amino acid will then attach to it. And this is just showing that growing amino acid here. This is also just kind of summarizing everything that I've said. After mRNA has left the nucleus through the nuclear pore, it will go to the cytosol, specifically the ribosome that's in the cytosol, and it will attach to it. The tRNA is going to bring over the amino acid that the codon codes for. The way that it recognizes it is by its anticodon that's found on the tRNA. And those amino acids will continue to join together, and they're joined together by peptide bonds, and it continues and continues until it reaches a stop codon. And so that is basically protein synthesis. I'm going to write what we call the central dogma of molecular genetics here. We have DNA to RNA, and then we have RNA to protein. This is translation, and this is transcription. This happens in the nucleus. This happens in the cytosol, but specifically the ribosome. And so we just talked about how it happens in the ribosome that's found in the cytosol. And so in order for it to attach to the ribosome is also made up of RNA. And we call that ribosomal RNA. And it has two subunits that help to bind to the mRNA molecule, and it allows that whole tRNA molecule to bring the proper amino acid to that polypeptide chain. And again, it continues and continues until it reaches a stop codon, and then it will stop. All right, this is one last time to practice about with protein synthesis. If this first chain was, or this first strand was a strand of DNA, I want you to write the strand of RNA, being sure to separate it into codons. And once you've done that, then use the chart that's in your notes to write out the polypeptide that is formed. Again, you should pause the video at this point. I'm also going to pause for a few moments. And so the RNA strand separating the codon should look like this. And the polypeptide form, which means we're taking this and looking in the codon chart to see what are the amino acids that are formed. Again, if you need to pause the video again, you can do that. But those would be your amino acids. What's so special about those amino acids? Hopefully you realize this is methionine, which is actually the start codon. And I picked these randomly, so of course the start codon would come before any others that are going to be part of the real protein. Okay. And one of the last sections we have here is changes in genetic information. And this happens quite often, just depending on the person, what they're exposed to. Now, most of our genome is going to be basically the same, but we do have some things that are going to vary. And it could affect a variety of things, like our appearance or health, or it could just not be any type of effects at all. The first thing we're going to talk about will be mutations. And with mutations, mutation itself is simply a change in a mutation is going to be a change in mutation is going to be a change in the DNA where it could be where something is added, deleted, or just changed in some way. And those can be spontaneous or it could be induced, which means spontaneous, something that we really can't control at all, or it could be induced, whereas we've been exposed to maybe smoke, we've been exposed to radiation, some type of chemical, it will cause that change within the DNA sequence. Now, some mutations may not be harmful at all, and we may not even see it, but some can affect health. For instance, some muscular dystrophy could happen. Also, another example would be a change of the gene of the hemoglobin molecule that could cause sickle cell. But then you have some mutations that become that are an advantage to your health. For instance, there is actually a mutation that people may not realize they have that protect them from HIV, where the receptor that virus binds to is incomplete. And so that is definitely a benefit. I would not want to test it out to see if someone has it. However, it is definitely an interesting mutation, and it's definitely one that is advantageous. And one of the last things is how we can kind of bounce back from mutations. So some mutations can be repaired, and it may or may not. And we also have, if you remember in the codon chart, where certain codons can specify that same amino acid. So for instance, if UAA, or shouldn't use that one because I think that's a stop codon, if CCC codes for one amino acid, but CCG also codes for that same amino acid, you won't have a difference in the protein at all. The difference comes where that one amino acid is changed, or that one nucleotide, if it is changed, it could possibly change the amino acid. All right, and that is it for this chapter.